Hi students, here is the narration for our study of the cardiovascular system, which includes blood, the heart, and a discussion of blood vessels. This narration will focus only on blood, its composition, essentially, and so again we will be looking at how the different components in blood serve some function in the body. So this image here shows a single drop of blood contains millions of red blood cells, white blood cells, and platelets. And one of each type is shown here. So here we have a red blood cell, which is often described as a biconcave disc, or sort of looking like a donut, except you don't have that hole in the middle. The middle is sort of pressed in. So it's as if you took a ball and you press the two sides together. And that's a very unique shape for a cell, and it is actually related to the function of this cell, which is to transport oxygen. More about that later. The other cell that we see here is a white blood cell, this one that looks like a ball with lots of things on the surface. And that's actually a good reminder that on the surface of our cells, embedded in the plasma membrane of cells, are many different molecules, and some of these molecules function as markers. But that's what's referred to as a white blood cell. The correct term for these cells are shown in brackets. So red blood cells are erythrocytes, and that prefix eryth always refers in medicine to red. Not necessarily red blood cells, because if you have a rash and it's very red, it's described as an erythema. But that prefix is what the eryth part means, red. Leuc, white. Cyte means cell, leukocyte, white blood cells. And we have many different types of white blood cells, while we only have one type, shall we say, of red blood cells. So they tend to be more circular. We'll have another closer look at them. And then the third structure in this diagram is actually a cell fragment. It is not a true cell. And that is a platelet. So platelets are absolutely needed despite their small size and despite the fact that they are not a cell. They are needed for blood clotting, and that's their function. So platelets are called thrombocytes, and again, that prefix thrombo in medicine always refers to platelets. So someone might have thrombocytopenia. That usually means a low platelet count, as opposed to a low red blood cell count or a low white blood cell count. So learning some of these prefixes can help you, figure out information later on in your studies. So please make sure you know the correct term for the cells as described here. Now for a closer look, let's see. If I were to take a sample of blood from you, as they do when you go to a lab, in the lab they put your blood into a test tube that has an anti-clotting agent. But if I put your blood into just a regular lab test tube, a smaller one, and I leave it on the benchtop, the cells and the platelets would settle to the bottom of the test tube. And because red cells outnumber white cells, the bottom of the test tube would actually look red. Red blood cells outnumber white blood cells in a drop of blood about 100 to 1. So there's always many, many, many more red blood cells than white blood cells in any sample of blood in a normal, healthy individual. Now the liquid portion of blood would be at the top. And that liquid portion is called plasma. So we have plasma, then we have a portion that contains only red cells, and between these two is what is called the buffy coat. And the buffy coat is where we find white blood cells and platelets, and you can see that just as a very narrow band, sort of whitish in color there. There's another name given to the percentage of red blood cells that are normally found in a sample of blood, and that term is called the hematocrit. And here's another prefix for you to remember, heme. Anytime you hear heme, you can also think of red blood cells. In particular, heme comes from the word hemoglobin. And hemoglobin is the protein found in erythrocytes that carries oxygen for us. So in terms of the composition of a sample of blood, this is what we have here. Roughly 55% is plasma, and the other 45% is the formed elements. So formed elements is a term to know. It's here in bold, and it comprises what you might want to think of as a solid part of blood, the cellular elements, but remember platelets are cell fragments. So erythrocytes, leukocytes, and platelets make up the formed elements of blood. So the formed elements then are the living component of blood. Plasma is the fluid in which the formed elements are suspended. Now, most of blood plasma is water. 90% is water. So if someone is dehydrated and you took a sample of blood from them and did exactly this, let the formed elements settle, you would actually get a much smaller volume, perhaps like that, of plasma. So we do need to be adequately hydrated in order to have enough plasma in our body. And plasma is very important. It is what helps the suspended cells travel around the body. What else do we find in plasma? Well, there are some proteins. And these proteins, and it's not protein, it's proteins, because there are different ones in there. They're referred to as plasma proteins. So an example, I'll only give you one example, but an example would be antibodies. We were all talking about antibodies during COVID. Well, antibodies are proteins. They're not carbohydrates, they're not lipids, they're proteins. So those proteins are actually suspended in plasma. So an antibody is a Y-shaped protein. So I'll draw one there, but we have millions in our plasma. So that's an example of a protein, and there are a few others. We also have some nutrients. So while this says sugar, that should really say glucose. That's blood sugar, glucose. We also have amino acids, which we remember are the building blocks of proteins. In addition, we have electrolytes. So electrolytes, these are ions. We've learned how important sodium ions and potassium ions are for nervous system conduction. We've also got chloride ions. And in addition, we also learned that hormones travel in blood. So we can expect to find some hormones in blood. So when a physician requires, you to provide a sample of blood, and they often have a sheet to accompany the request, and on the sheet, they check off a long list, many things that they can look for. Check your glucose level in blood, check your creatinine level in blood, and so forth. And they can ask, for a complete blood count. They can ask for the hematocrit. So a sample of blood actually gives a lot of really useful information to a physician about how well certain organ systems are working. And it certainly is part of the criteria, for example, for diagnosing whether someone has excess blood glucose and therefore they are now going to be classified as a type 2 diabetic, for example. So this statement here, a sample of blood spun in a centrifuge. A centrifuge, I mean, you can look it up, Google what a centrifuge is, but it's an instrument that's often used in labs. It has a very fast revolution, so it allows you to separate substances that are present in a liquid rather quickly. You know, you spin something in a centrifuge and it's done in seconds or minutes. Okay. So depressed and elevated hematocrit levels are shown. So here is someone who might be anemic. We probably all have heard this word, you know, if you're anemic. Bear in mind that there are different types of anemia. So, but the word anemia is kind of the umbrella term for many of those different conditions, but they're all related to the fact that since red blood cells carry oxygen to tissues and cells, if we're anemic, we do not have the oxygen arriving at cells. It could be that we're not producing enough red blood cells. It could be that there's a problem with hemoglobin, because hemoglobin requires iron to be made. So if our iron levels are low, we also require the vitamin B12, for example, to help in the process of red blood cell production. So they're usually, if someone is anemic, the physician has figured out exactly which component that's required for red blood cells to do their job, which one is malfunctioning. But this is one way to determine if the hematocrit percentage of red blood cells is lower than expected. And so here we have for this symbol, the first symbol here is for females with a little cross at the bottom of the circle, 37% to 47% hematocrit in a sample of blood. That's a normal level. So if you are anemic, you have less than 37%. And if you're male, it's 42 to 52. And this difference is due to the difference usually in body size and composition. 42% to 52% hematocrit. The opposite problem could be you have something called poly. Poly again means many. Polycythemia, too many red blood cells. And this could indicate a hormonal problem or other types of problems. Now, this word we met when we were studying the skeletal system, hematopoiesis. And hematopoiesis refers to the formation, the production of all the cells in blood. So all the erythrocytes, all the leukocytes, and the platelets. And this process takes place in red bone marrow. So again, not yellow bone marrow. And we remember red bone marrow is found within spongy bone, usually of long bones. Now, this is a process. Hematopoiesis is a process, which means it has a beginning and it has an end. The beginning starts with what is called a stem cell. And by definition, a stem cell is a cell that can give rise to many different cell types. I do believe I've said this before, but scientists have been working for the last 20 years at least with stem cells because they are able to take stem cells, put them in a petri dish, and basically grow up, for example, an organ like a kidney. That's how far science has advanced. The question is, why would they want to do that? Well, because there are never enough organs around for people who require an organ to be donated to them to save their life. So this is seen as a way around that problem. But back to hematopoiesis. There are two types of stem cells here that are shown here, but I would like you to know that there is an original cell, which is also a stem cell, and that cell divides to give what is called a myeloid stem cell and a lymphoid stem cell. These are important words to know, lymphoid and myeloid, because they sort of represent branches. Think of it like a family, branches of the family tree. So here's the original parental cell, which is a stem cell. And then these are like the two sort of daughter cells. But one differentiated along this pathway. And when I say differentiated, it usually means that the stem cell was subjected to a variety of biochemical substances in the body, in the bone marrow. So this one differentiated along one pathway, and this one differentiated along a different pathway. So right now, I've got the Olympics on my brain, so maybe this one here decided he wanted to become a downhill skier. And this one here decided to be a hockey player, you know. But both of them have the potential to differentiate further. So the lymphoid stem cell differentiates to give us what are called the agranulocytes, small lymphocytes. And these are really T cells and B cells. You can barely see the T here, so I'll brighten that up. Something called a T lymphocyte and a B lymphocyte. I use the word cell because I guess it was just easier during my training, which was in the area of immunology, for us to not say the long word lymphocyte when we're chatting. We just say T cell and B cell. Of course, in talking to other people with similar training, we all knew what we were referring to. This also shows a very unusual cell called a natural killer cell. And a natural killer cell doesn't appear anywhere else on any of my slides. But this cell was actually discovered during all the research that was being done for cancer. And it was found that this is actually a cell that bears many similarities to a lymphocyte, but it's not identical. And it's very, very different from the other types of white blood cells. And because it actually could kill cancer cells, it was labeled or named a natural killer cell. So that's its most, its claim to fame. The natural killer cell, a cell that can kill cancer cells. And while we're on the subject of killing, you need to be very careful with how we sort of use that word. When we're discussing the immune system, which is coming up, because we're going to be talking about these cells again when we discuss the immune system. There's a type of T cell, T lymphocyte, that is able to kill infected cells. So these are the only two cells, the natural killer cell and the T lymphocyte, what is actually called a cytotoxic T cell or a T killer cell. Those are the only two that can actually kill another cell. B cells do not do any killing of any kind. Neither do these. So these are the final differentiated cells from the stem cell that went in a different direction, matured in a different direction, a different lineage, the myeloid stem cell. And these various intermediates, do not try to memorize them. That's not the expectation at this level, but it's just to show you that it's a process. And so it's as if all of them have, one original grandparent, but then they all kind of went along their own way. So this is the precursor cell for this one, the megakaryocyte. And a megakaryocyte is a cell that does nothing that we will discuss. Its job is to produce platelets. A fragment of a megakaryocyte is actually a platelet. Then there is another progenitor cell that differentiates to give you the erythrocyte, the red blood cell. Then there is another one here, something called a myeloblast, that differentiates to give you 3 different types of cells, all which have the ending "phil" in their name. Basophil, neutrophil, eosinophil. Watch out for these. They will come back in our discussion of the immune system. And then a monoblast gives you what is called a monocyte. So this slide is on hematopoiesis. So understand that it's the process of making and maturing. So all these cells at the very end of the process are what we call the effector cells. They all have a unique and specific job to do. Blood is the medium that transports them around the body. But these cells are really part of our immune system. The only exception is platelets, because platelets, they only participate in blood clotting. But blood clotting, which is, part of like repair after injury, is also part of an immune response. Red blood cells here also, their job is to carry oxygen around the body. So the basophil, the neutrophil, the eosinophil, the T cell, the B cell, and the monocyte. They are all big players in an immune response. A closer look at erythrocytes. So the shape of erythrocytes allows them to do two things. They can stack the way you would stack your plates in the dishwasher, right? Because if you stack them, like this, then they would stack very neatly, especially if your plates are all the same size. So red blood cells can stack like this, and that allows them to pass through blood vessels, because we have a lot of red blood cells in a drop of blood. Additionally, we also have a lot of oxygen being transported by these red blood cells. The other thing that this shape allows the cells to do is that they can fold. So because of a flat center, they can fold over, and that allows them to move through narrow blood vessels. So the blood vessels that we're going to talk about in the next lecture, the next topic, would be arteries, veins, and capillaries. And of those three, capillaries are the smallest in diameter. A capillary can have a diameter that is about 1/10 the width of a strand of hair. And these cells have to be able to pass freely, easily, and many of them have to be able to pass rather quickly. What is on the inside of a red blood cell? Well, when you learned about the eukaryotic cell, you learned that the cell had a nucleus. Outside of the nucleus, you had the rough endoplasmic reticulum. You had a Golgi apparatus. The cell might have lysosomes, mitochondria, smooth endoplasmic reticulum, etc. That was the typical eukaryotic cell that you studied in Bio 1150. Well, in the process of moving from being, the stem cell that's going to produce a red blood cell, all of those things are removed. A red blood cell does not have a nucleus, does not have rough endoplasmic reticulum, no organelles, not even mitochondria. So it's an example of a cell whose structure has been modified to suit its function, because instead of all those things which a red blood cell does not need, it has this protein, hemoglobin. Since oxygen is needed for life, because oxygen plus glucose combines in cellular respiration to give you carbon dioxide, water, and ATP. All of the oxygen that we inhale is used by the body for that reaction. It is not used for anything else. And all cells, with the exception of red blood cells, carry out cellular respiration. Red blood cells, they have the job of transporting all of that oxygen to every single cell in the body that needs it. And some organ systems use more, like the brain. The nervous system uses far more oxygen than other organ systems. So, hemoglobin is the protein that can bind oxygen. But hemoglobin can also bind carbon dioxide, and it can also bind carbon monoxide, which unfortunately is why carbon monoxide can kill us. Because when carbon monoxide binds to hemoglobin, oxygen can't bind. Without oxygen, cells die. So a red blood cell is really like a sack filled to capacity with hemoglobin. That's basically what it is. So a single erythrocyte can contain 300 million hemoglobin molecules, and each hemoglobin molecule can bind 4 oxygen molecules. So they can transport, this is a single erythrocyte can transport 1 billion oxygen molecules. There is no other cell in the body that can perform the function of red blood cells, which is why if your red blood cell count is low, your hematocrit is low, or your iron levels are low, because hemoglobin has to have iron in it. Iron is part of the structure of hemoglobin. You feel tired, you have no energy, etc. Those are some of the symptoms that people have when they're anemic. But that's the reason why. So even though you probably knew that red blood cells were very important, I hope you have a better understanding of why, from a scientific point of view, why they're important. Now, since they do not have a nucleus, they have no DNA, they cannot undergo mitosis. So they cannot replicate themselves, which means that the body has to continuously produce red blood cells on a daily basis. So red blood cells are produced millions every single day. So in this diagram, that's just showing you some of the sites, where we can expect hematopoiesis to be going on. Now, since they cannot undergo mitosis, that means they have a finite lifespan. So they live for approximately 4 months, and then they die. But instead of all that iron being wasted and going out of the body as waste, it's actually recycled and used to make more red blood cells, because we have to produce so many millions per day. And the other part, so what this diagram here is showing you is, this is the structure of hemoglobin there. So hemoglobin is made up of 4 globin chains. The protein is globin, and 4 globin chains have to come together and form the structure. So if you remember, proteins have a 3-dimensional structure, a shape that makes them functional. Hemoglobin has a quaternary structure because it requires 4 different globin chains to come together. Individually, these globin chains are non-functional, like here. So when the red blood cell is kind of old and broken apart, the globin chains are separated. They're broken down to amino acids, which the cell can use to make proteins. And the heme part, which is where we find the iron atom, that is reused by the body to make more hemoglobin. So that's why this is kind of a kind of recycling going on here in the body. And it's actually a really beautiful process. The liver is involved, which is why the liver is shown here. Now, I'm not saying that you have to memorize this diagram. I would say you don't have to, but try to understand and appreciate what the diagram is trying to tell you. So the liver, at the end of the life cycle, they are destroyed in the liver, though some of the components, such as hemoglobin molecules, are recycled and used again. Now for the white blood cells, the leukocytes. Now you can always remember, of all these leukocytes, in a sample of blood, which ones, again, we're talking about a normal, healthy individual, which of these would be most abundant and which one would be least abundant. And so there is this little statement that you can memorize. Never let monkeys eat bananas. And the first letter refers to the names. So neutrophils are the most abundant in any sample of blood. Lymphocytes are next. Monocytes, eosinophils, followed by basophils. So take some time to look at the image of these different white blood cells, because on a blood smear, so if you had a microscope or if I give you an image of a blood smear, you ought to be able to recognize these. The dark staining purple in all the images represents the nucleus. And so one of the interesting things that you will see is that the nucleus is not this circular, ball-looking structure that, again, you were led to believe when you studied the eukaryotic cell, that all cells may have a nucleus that looks like that. These cells are actually recognized both based on the color, because when you make a microscope slide, what you do is you would take the slide, which is a piece of glass, you will take the drop of blood, put it in the middle, then you will take another slide and you will smear it. The reason you smear it is that whenever you're doing microscopy, you want the light, because the light comes from the bottom of the microscope, you want the light to be able to pass through the slide because your eyes are here. And if the light can't pass through, you won't see anything. So you smear it and then you add dyes. You stain the sample is what we call. So you stain with dyes. And the reason we use dyes again is because with the color from the dye, we can actually see our specimen rather easily. Without a dye, it can be very difficult to see your specimen, even though you've got enough light coming through on a microscope. So the type of stains that were used here stain the nucleus for these cells. So this is not, looking at the neutrophil here, this is not a couple of nuclei. This is one nucleus, but it has lobes to it. So a neutrophil is described or recognized as having 3 to 5 lobes in the nucleus. Whereas a monocyte tends to have this U-shaped, large, it's a larger cell, a U-shaped, again, dark purple staining nucleus. Lymphocytes tend to have, the overall cell tends to be smaller in comparison, let's say, to a monocyte. A monocyte is one of the larger cells of the white blood cells. A lymphocyte is much smaller in comparison, even though this image isn't saying that. But from a practical perspective, I can tell you that. But you recognize a lymphocyte because the nucleus is so large, it does tend to be circular, and it tends to take up almost the entire cell. The other interesting thing about these two is these two get labeled as agranulocytes. Why? Because if you look at the basophils, all the phils, basophil, eosinophil, and neutrophil, their cytoplasm, these what looks like little dots in here, these are little sacs of granules, vesicles of granules. And remember that a long, long time ago, more than 100 years ago, when these cells were discovered, when we had microscopes that now allows us to see things from the body, and scientists were trying to figure out all of this, they had to name some of these things. And they also sometimes wanted to have names that would help them to remember what they look like, or that the names were descriptive. So these three fall into the category of granulocytes because they have a very granular, their cytoplasm looks like it has grains inside That's where the word granular comes from. Same with this one here, the basophil. And what you're seeing actually, the little pink, these circles here, these are erythrocytes. And you see this erythrocyte here, how it looks as if it has like a white dot in the middle. That's again because of the biconcave shape. When you put it flat on a slide, because these are images from a slide taken via microscope, this is the light is shining right through. I'm looking to see if I can see a platelet somewhere in here, but I don't. So we have the granulocytes, which are the phils, basophil, eosinophil, neutrophil, and then we have the agranulocytes, which is the monocyte and the lymphocyte. These are just ways of classifying the white blood cells, the leukocytes. So watch the word leukocyte and lymphocyte because they don't mean the same thing. A lymphocyte is a type of leukocyte. So now we look a little bit closer and now you can see the granular white blood cells. So the neutrophil is the most abundant. What is the job of the neutrophil? It functions as a phagocyte. So what is a phagocyte? A phagocyte is a cell that basically can chew up bacteria. So the cell, so let's take this neutrophil here. If there was a bacteria out here, the plasma membrane actually circles around and brings the bacteria in. Now these cells, these white blood cells, they have none of the characteristics that we said, you know, a red blood cell. Remember, a red blood cell had no nucleus, no organelles, etc. These white blood cells, they are like your eukaryotic cell. So they have nucleus, they have endoplasmic reticulum, etc. They're what you might think of like a normal cell would look like. So they would have lysosomes. And if you remember, a lysosome was a small sac, a vesicle, that contained digestive enzymes. And so the bacteria would be brought in and the digestive enzymes can destroy the bacteria. The neutrophils would probably die most often. So if ever you see pus, P-U-S, around a wound, usually that's dead neutrophils, phagocytes. Now, the other two, eosinophil, this one's claim to fame is that it tends to be predominant if you have a parasitic or a worm infection. So, you know, you can get parasites from eating food that isn't cooked properly, drinking certain types of water, etc. Basophil, this one here, you want to pay attention to. It has large granules that stain dark blue to purple with a two-lobe nucleus. So this one, basophil, eosinophil looks more like earmuffs. Basophil, at least in this diagram, you can't really see the two-lobe nucleus. It sort of looks more, I don't know, like an elongated nucleus. But basophils play a role in allergic reactions. And you should remember this, that basophils have histamine. So all of these little blobs, these little sacs that are shown here, they contain histamine. And histamine, we will talk about probably a little bit more in the immune system, because histamine is what triggers what is called inflammation. And so an inflammatory response has a particular pathway that it follows. But one of the symptoms, or the symptoms that we see, or a consequence of this, you know, inflammatory pathway is this: redness, itchy skin, and watery eyes. So think of anybody who has seasonal allergies, allergies to pollen, they get hay fever. Hay fever is the result of an inflammatory response. And the cell that's responsible is this one, the basophil. The same thing is true if you are allergic to food, peanut, etc., or you are allergic to bee stings, and so forth. So Of these three, neutrophils are usually at work in the body because, if you're healthy, it doesn't mean that you are not getting bacterial infections or maybe even viral infections viruses entering your body. What it does mean is that even if they enter your body, your immune system is very robust and it is able to shut down any possible infection before it becomes overblown and make you feel sick. So the A granular white blood cells, monocytes and lymphocytes lack grains, as we said. Monocytes are the largest of the leukocytes, and monocytes can undergo some changes, and they also function as a phagocyte. I'm going to try and spell that word for you. It's P-H-A-G-O-C-Y-T-E. And this prefix phago comes from the word phage, which means a cell eating another cell. So that's how it got its name. Lymphocytes are the second most abundant of the leukocytes, and the size of the nucleus takes up most of the space in the cell. There are two main types, B and T cells. Lymphocytes are important in creating antibodies. Let me just clarify, it is only B lymphocytes that make antibodies for us. No other cell, no other lymphocyte, okay? So they're a very important part of our defense against infections. T cells help, but you will have to wait until we probably get to the immune system to have a better understanding of how T cells work. But T cells, T as in Tom, T cells do not make antibodies. So you can go through the learning check and then check in with me. The last one here, thrombocytes, we said were a fragment of a cell. They come from a cell called a megakaryocyte. And so platelets are required for blood clotting. So, we've all experienced this. You get a cut, you get a clot formation. The function of a clot is actually to seal the opening of the wound, because if the wound is open, then obviously bacteria, virus, infectious agents can get into not just the wound but into your body that way. It's part of the reason why you also want to cover an injured site in the body. Okay. Clotting helps to prevent blood loss. This is a second function. It also helps to prevent blood loss at the site of injury or within the body. So when a clot is formed, usually eventually it becomes a scab, and you probably know that if you take that scab off, it starts bleeding all over again. So what is required for clotting? Well, a number of things. First, you need platelets. So that's number one. You also need calcium ions. Number two, which I think I mentioned when we were talking about the endocrine system and why calcium ion concentration in blood needs to be regulated by two hormones, calcitonin and parathyroid hormone. The third one is one of those plasma proteins, fibrinogen. Now, any name that you find in biology that has an O-gen as part of the ending, it means that it is inactive. So fibrinogen is circulating in blood because it's one of the plasma proteins, but it is not active. So it has to be activated in a process. So fibrinogen is converted to something called fibrin. And fibrin are protein strands that help to trap red blood cells to form the clot. So you might want to Google image of a clot, but what you will see is something like this, where you have fibrin strands and cells. Most of the time, again, you'll see red blood cells because they're the most dominant cell type in a sample of blood. but the cells are trapped in there, and that sort of mesh, solid, formed thing is your clot. So platelets are the least abundant formed element in a blood sample. Now we get to something very interesting, something that sometimes leads to a lot of confusion with students. So I would say that take your time going through this, and we can discuss further at one of our meetings. And this is the subject of ABO blood types. So I think all of you know that you've all been assigned a blood group or a blood type. The ABO blood type is not the only one present in humans, but it is the one that causes the most problems, usually medically. And I think we all know that, the Canadian Blood Services constantly ask for blood donations, because when people are having surgery and certain medical procedures, etc., they may require blood to help them recover. So the ABO blood group, there are 4 blood groups. You can either be blood group A, type A, blood group B, type B. You can be type AB, or you can be type O. Those are the 4. And if you don't know your blood group, because you should, you can check your medical record or ask your doctor, because they definitely know. So the question is, how does someone get assigned to a blood group? Usually you're assigned at birth, at least in Canada. And how do we know? Well, a sample of your blood is taken to a lab and it is typed. And what they're looking for is this. On the surface of red blood cells, there are 2 molecules. Those molecules are referred to as antigens in medicine. All right? Because molecule is too general. So they're referred to as antigens. And to keep the naming simple, they simply called one of them A antigen and the other one a B antigen. They have different structure, so that's why they both couldn't be A or B. So on the surface, so here's your red blood cell. If, let's say, I'm going to use a triangle to represent the A antigen. This is a red blood cell. So this discussion is all about red blood cell, nothing to do with white or platelets. So that is the A antigen on the surface of the red blood cell. It looks like an A. Let's draw another red blood cell, and this time I'm going to draw something that looks like a B. And that's the B antigen on the surface of a different red blood cell. So if you only have the A antigen on the surface of your red blood cell, you are put in this group, type A. So everybody who's type A has the A antigen and only the A antigen, or the A molecule. If you have only B, you're put into this group, type B. If you happen to have both, because some people can, so that means you would have this and you would have this one. I mean, I've drawn them as if they're touching, but they don't have to be touching or close by. That would be A and B. So this group is called AB. So everybody who's group AB has both of these antigens on the surface of their cells. Again, red blood cell. And then there are individuals who have neither A nor B on the surface of their red blood cells. They are put into blood group O. This is important to note. There is no such thing as an O antigen, which I've seen students write on a test. So O, think of the letter O looking like the red blood cell, but nothing on the surface. Now, when we are born with all of this. A antigen on the surface of our cells, B antigen, or both A and B, or not, neither. But in addition to our red blood cells, there is something else that is found in our plasma. And the something else that's found in our plasma is called antibodies. It's important to remember that we are born with these antibodies also. Now, here is where students tend to make their mistakes. If we are born with type A blood, which means that we have the A antigen, we do not want the A antibody. Because normally, whatever this antigen is on the surface of a cell, here is the Y-shaped protein, that's an antibody. If this is A antigen, the A antibody can bind to it. But when we are born, we don't want this. Why? Because if this happens, the red blood cells are destroyed. Antibodies are part of an immune response, and usually when antibodies bind to things, in the case of red blood cells, you get this agglutination, clumping of the red blood cells. So clumping is not clotting. It means a lot of cells get joined together. The effect is very similar to clotting, but it is not clotting. It is called agglutination. But it's the same problem like a clot. A clot could plug up blood vessels. If it plugs up a blood vessel to your brain, well, brain doesn't get oxygen. If it plugs up a blood vessel to the heart doesn't get oxygen, the muscles won't get oxygen. Those are very critical situations, life and death situations. So the key to understanding this is that an A antibody can only bind to the A antigen. That's how antibodies work. Antibodies have defined specificity. The B antibody cannot bind to the A antigen. So since the B antibody cannot bind to the A antigen, think of it, it cannot see, right? Because the B antibody would not, this is the part of the antibody molecule here that basically sees and binds to this, the antigen. But the B antibody now will look like this. This is what the binding site would look like for the B antibody. This shape is very different from that, and it cannot see it, it cannot bind to it. So it is safe for somebody to have B antibodies in their plasma. Similarly, if you're type B, you do not want the B antibody, you want the A antibody in your plasma. If you have both the A and the B antigen here on the surface of your red cells, you cannot have either the A antibody or the B antibody in your plasma. So you have neither one. And if you're type O, well, you don't have any antigens on the surface of your red blood cells, so it's okay to have both antibodies in the plasma. So take the time to digest that, because this is very important information to learn. And so here on the slide, this image is meant to show, for example, this is a red blood cell from somebody who is in blood group A. And so this is meant to represent the A antigen. And you see the A antigen has a different shape or a different structure from the B antigen. So this is representing somebody from type B. This is their red blood cell. And if you are AB, you have both the A antigen and the B antigen. And if you're type O, you do not have either A or B on the surface of your cell. So antibodies, so let me explain this because I just told you that an antibody is a Y-shaped protein. This would represent one arm of, this is a very, in this image, they chose to show you an antibody that's called IgM. IG, okay, IG means immunoglobulin, and immunoglobulin is the correct term for antibody. And this particular antibody that they're showing here has five, it's called a pentamer. It sort of has 5 arms to it. It's a very large antibody. Not all antibodies look like this. But nonetheless, these are the binding sites on the antibody molecule. So this one is shown in green to represent the anti-B antibody. I was calling it the B antibody because that was on the slide, but the correct way to refer to antibodies, so be very careful with the words antibodies and antigens. They don't mean the same thing. They're like opposites. So, and the antibody is named anti-B antibody. This is the anti-A antibody. So while it looks very similar, they've used a color here to show the difference. But if you could actually see this, like at the molecular level, if you could see this, what you will see is the shape of the binding site, these 2 sites right here, is very different from the shape here, between an A and a B. And obviously for AB, you can't have any. Why? Because if you have the A antibody here, if you had this here, A antibody would bind to the A antigen there on all the red cells. It would cause agglutination clumping. So in the body, we do not want agglutination. And this is what is needed to be avoided when someone is receiving blood. The recipient cannot receive blood that will cause agglutination in the recipient's body. Okay, now a few more slides to go. How to type a person's blood. Blood typing is a process that's done in a lab. Blood typing is not blood transfusion. Blood typing is figuring out what the person's blood group is. Is this person group A? Is it group B? Is it AB? Is it O? So it's actually quite simple to do if you have the right stuff to do it with. But basically, this is showing you the result for one person, and this is showing you the result for another person. So we have these plastic or glass plates in a lab, and they have 3 wells. So this well here, if you look carefully, it's labeled A. And this one here is labeled B. So there's a B here. And this one here is labeled RH. Now I haven't said anything or discussed RH. There's no information about RH on the slide. What is RH? Well, I think we all know that most oftentimes when we say we are a blood group, there's always a positive or negative, right? We're either A positive or we're A negative, or we're B positive or B negative, AB positive or AB negative. So what does that mean? If you're positive, it means you have, in addition to the A antigen or the B antigen, you have another antigen called the RH antigen on the surface of your cell. So if you have it, the RH, you're considered positive. If you don't have it, you're negative. So because it's on the surface of the cells, we can detect it. So how do we detect it? Well, we use antibodies to detect all of these, A, B, and RH. So the first thing to do is take the person's blood and simply put it in this well, and this well, and this well. So the sample of blood is in all three wells. Now, what do we add to well A? Well, we add the anti-A antibody. What do we add to well B? We add anti-B antibody. And to RH, we have anti-RH antibodies. These antibodies that we're buying, we buy them from a company. They're companies that manufactured these. There's a way to do this scientifically, right? So we know what we're adding to the well. We know which antibody we're adding to the well. Now, if we get agglutination, what we see is kind of this, You see, when a drop of blood will remain kind of smooth looking, like this one is very smooth looking, whereas this one has got little clumps happening around here. So whenever we see little clumps, that means there's agglutination. And agglutination means that the antibody that you added was able to find its antigen and cause agglutination. So we add the A antibody. This is an antibody, AB antibody, RH antibody, B antibody. So we're adding antibody, and the red cells are here. And if the antibody can bind to the antigen on the red cells, you'll get agglutination. And then you know that, oh, that red cell has, if you get agglutination, it means the red cell has A. If you get agglutination here, it means the red cell has B. If you get agglutination here, you say that the red cell has RH. So no agglutination here means that you do not have the A antigen. No agglutination here, because you see smooth, smooth looking result. You do not have the B antigen. But here there's agglutination, therefore the person has the RH antigen. So what is their blood type? Well, the person who doesn't have A or B is type O, but since they have the RH, they're O positive. Exactly the same thing is done on this side. So where do we see agglutination? Here. There's a little bit of clumping there and there and there. So this person then, again, you're adding the A antibody to the A well, the B antibody to the B well, the RH antibody to the RH well, and you look for agglutination. Only these two gave you agglutination. So the person has the A antigen, so they must be in group A. They have the RH antigen, so they must be positive. So the person is A positive. So this is blood typing. Like I said, it has nothing to do with donation. But if you decide to go and donate blood and you don't know your blood group, or even if you know your blood group, they probably might not believe you because they want to be 100% correct when they label the bag that contains your blood with your blood group. So they will actually type your blood in a few minutes and have the answer. So they have a concrete, accurate, correct answer of a person's blood type. So like I said, this would take 5 minutes or less to do. So in diagram 1, which well shows agglutination, you can figure this out. And so there are some donation rules here. Only red blood cells are donated, so always think about who is the recipient and who is the donor. Okay? Only antigens are donated. A person who is Rh positive can accept both Rh positive and Rh negative blood. A person who is Rh negative should never be given Rh positive blood. Why? Because their body will make antibodies, anti-Rh antibodies. So antibodies of the donor are not donated, only antigens are donated. And there's a game that you can play. So I hope you enjoyed learning about blood, and see you again.